Hello, my name is Dave McCarthy. This is for medical physiology at the University of New England. Today we're talking about the determination of gender with genes and hormones. So let's get into it. Basically all starts with DNA. We have 23 pairs of chromosomes in the body, 22 autosomes, and the last pair are the sex chromosomes. We have the XX for female and the XY for male. As you can see down here, we're showing the XY, so this is a male. So genes, with the 22 pairs of chromosomes, they're considered diploid. They're the same, except for the sex chromosomes. They're haploid in nature. And the X, as we saw in the earlier photo and here, the X is a lot larger than the Y. Okay, so one set comes from the mother, here, and one set comes from the father, here. All right? So, sexual differentiation. So we basically have the male and the female, and within the fetus, as the embryo develops, we can see how the glands at one point in time shift. So the mother always contributes the X, and the father either contributes the X or the Y. All right? And in the female, only one of the X is going to be activated. The other one's going to be dormant. Okay? So at this certain stage in the embryonic tissues, there is encoded on the Y chromosome the SRY gene, which basically, if it's present, as in here, it will go towards the male sex. But if it's negative, it'll go towards the female sex. So, if the Y chromosome is present, it will go towards the nature of the male. Genital structures are different. The three, kind of broken up into three different areas. You have the gonads, and it's both the same. This is where the production of gametes happen. Either the sperm or the ovum. Okay? Sperm within the testes, ovum within the ovaries. Okay? And then we have an internal genitalia, which is usually glands and ducts for excretion. Okay? And then the external genitalia, which is basically external reproductive structures. Within the male development, we have the testes, basically. That's where the zygotes are developed. Okay, you have the cereoli cells that secrete an anti-Müllerian hormone. Okay, in order to inhibit the testes from forming into ovaries. And then you have the Leydig cells that secrete antigens like testosterone and DHT, okay, dominant steroid hormones. So basically the testosterone is going to be in charge of making sure it pulls and lowers the testes out, so that rather than be up inside like the female, they'll be external. And the DHT is there for all other male characteristics, okay, basically completely structural in nature. As for the female, since it has the absence of that SRY gene, the cortex develops into ovarian tissue, so it stays up inside, everything develops. The Müllerian ducts, okay, then they start to develop into the majority of the physical characteristics of the female's anatomy, as far as the vagina, the cervix, the uterus, and the fallopian tubes. So basically, in conclusion, genetics set the tone for the production of hormones. Once we have that genetic outline, then we can set up which hormones will be triggered or not triggered, or suppressed or not, or excited, in order to change the anatomy and get it structured in the correct locations and produce the correct type of gametes. Thank you for your time.